So the neurotransmitters that are released into a synapse are all synthesized by our presynaptic neurons and stored within vesicles inside our synaptic knobs. When the synaptic knob is triggered through an action potential, then that synaptic knob is going to release the neurotransmitter, and that's going to diffuse across our synapse and bind into our postsynaptic neuron. This is going to trigger some kind of physiologic response in the target cell, and we've classified up to about 100 different neurotransmitters. Acetylcholine, of course, is everybody's favorite neurotransmitter. It differs substantially from other neurotransmitters. Biogenic amines, such as the catecholamines like dopamine, are important. The indolamines like serotonin are also important. We have amino acids like glutamate and glycine and GABA that can be used as neurotransmitters. We have neuropeptides, which are just short chains of amino acids that can also be used as neurotransmitters, including endorphins, which make you feel really good, and substance P, which is what causes pain. We can classify neurotransmitters not only on their structure, but also by their function. They can either elicit excitatory postsynaptic potentials or inhibitory postsynaptic potentials. And here's where it gets really exciting. Some neurotransmitters can cause certain organs to be inhibited and other organs to be excited. And then we can look at classification by action. Direct action, the neurotransmitters will bind to the receptors and immediately start to change our postsynaptic potential. Indirectly, we're going to use what's called a G protein second messenger system. You should know acetylcholine. It's our number one neurotransmitter. It's everybody's favorite, of course. It's used in the peripheral nervous system to stimulate skeletal muscle. It's always excitatory on skeletal muscle. It's used in the central nervous system to increase arousal. It's synthesized from acetate and choline. Action potential triggers its release into the synapse. It's going to attach to ligand-gated cation channels, and then it's going to be broken down with acetylcholine esterase. Now, as we use our neurotransmitter and we're done using it, we can get rid of it in several different ways. Most of it's degraded by enzymes. We can also have our presynaptic axon terminals reuptake or reuse the neurotransmitter. They're going to absorb it, package it back up into vesicles, and then they can release it again. All of our drugs that are psychotropic are going to affect the normal transmission of one or more neurotransmitters in certain areas of the brain. For example, selective serotonin reuptake inhibitors, or SSRIs, are used to treat depression. Selective means it is only used in certain areas of the brain, and serotonin reuptake inhibitor. So it prevents serotonin from being reuptaked and traps it in the synapse, causing it to stimulate our postsynaptic neuron again and again. Galantamine hydrobromide is an acetylcholine esterase inhibitor that we can use to treat Alzheimer's disease. All these drugs, medications, illicit or recreational drugs, will all affect the brain by interfering with the normal process. If the person in question is within homeostasis and their processes are normal, then these drugs will knock them out of homeostasis. Whereas if someone is outside the normal range, say they don't have enough serotonin within their synapse, by forcing it to stay there longer, then you can bring them back up into their normal range. Neuromodulators are chemicals that alter the response of local neurons. Facilitation is modulation that causes a greater response on our postsynaptic neuron. Inhibition is going to decrease the response in our postsynaptic neuron. Nitric oxide might be a neurotransmitter or a modulator. It's a short-lived nonpolar gas. It's made and released in the postsynaptic neurons in the brain, where it's believed to help strengthen memory by affecting presynaptic cells. The endocannabinoids influence the same receptors that marijuana does, hence the cannabis. Endo means from within, so the endocannabinoids are made by the body. These are small nonpolar molecules. They're made and released by our postsynaptic neurons, and they have an effect on presynaptic neurotransmitter release, and they help to influence memory and appetite. Now, I want to talk about neural integration and neuronal pools in the central nervous system, also known as neuronal circuits. These are groups of neurons that are arranged in specific patterns. Typically, their circuits are going to either converge or move together, diverge or start to separate, reverberate, which is bounce back and forth, or be parallel after discharge. Pools can be local, or they can be distributed in several regions of the central nervous system. So here's a converging circuit. We've got several neurons all converging or meeting on one single neuron. We'll have multiple sensory inputs on, say, the salivary nucleus. So sights, sounds, smells of cooking are going to all lead to one output, and that's salivation. Diverging circuit's going to spread information. We're starting with one or just a few neurons, and we're spreading that signal out more and more. Neurons in the brain that control walking are going to send commands to several different muscles for proper balance, posture, and motion. So we've got one small area of input, and you're causing different effects that are almost pre-programmed out in the muscles. A reverberating circuit's a little more complicated. We're going to use feedback to reproduce a repeated cyclical activity. So this goes in cycles. Once started, it stays there until it's interrupted somehow. We use these for circuits that keep us breathing while we're asleep. And then parallel after discharge is input transmitted simultaneously along different pathways and all end up at the same output. Since the pathways vary in length and the number of synapses, all of these signals are going to arrive at various times on the final neuron, and we believe these are used in higher order thinking.